ecological communities change. Species that were on Earth millions of years ago in the ecological community is not what we have today. And typically, big changes in ecological communities are coincident with major climatic changes. In the past, many of these climatic changes occurred because of big geological events. So for example, millions and millions of years ago, there was this massive volcanic or geologic event over Siberia called the Siberian Traps. It released tons of carbon dioxide into the atmosphere, bringing about climatic change. And so as we add more carbon dioxide to our atmosphere today, the question that others, ecologists like me, wonder about is what the ecological communities of the future are going to look like. How is this going to impose change? We have some insight into what communities of today look like under high CO2 environments from natural experiments, again caused by volcanic events. So this is a study off of Italy, kind of close to Mount Vesuvius. And because Vesuvius is still active, it's causing, these are bubbles of pure carbon dioxide that are coming out of the sea floor because of the volcanic events. And what this has done is basically created an experiment of what an acidified, or a community looks like in an acidified environment versus an area that's not affected by what they call a champagne study. And what you can see is that there's a lot of life in both locations, but the communities are very different. So here, in the community that's not influenced by carbon dioxide bubbling, you see a lot of calcifiers, a lot of diversity within the animals. So calcifiers are things like the urchins. The pink in that image is a type of algae that produces calcium carbonate. And you can see probably some corals back there too. But what you see in the high CO2 site, where the champagne site is a lot of primary producers, so a lot of algae. So again, an abundance of life in both areas, but very different communities. And this is something that ecologists can look at and say, this can give us a window of how communities might change with increased carbon dioxide emissions. And there's a number of these sites around the world. There's some, I think in Borneo, there's one in Baja, California, I think two in the South Pacific. I'll go into a little more detail on how and why organisms are responsive to carbon dioxide. So you can do a natural experiment with yourself. If you want to, hold your breath, and eventually you'll feel the urge to breathe. Most people think that's because you're running out of oxygen. It's actually the opposite. You're building up carbon dioxide in your body and your tissues. And what your body is doing is sensing the drop in pH caused by the buildup of carbon dioxide and telling you that you're in trouble, that you need to breathe out, you need to release this acid gas. And as Dick referred to, acidosis is a major health problem that's typically linked with problems of respiration when you're not able to release the CO2 from your body. There's a variety of molecules that are changing in ocean chemistry because of the invasion of carbon dioxide. So we have carbon dioxide itself increasing, hydrogen ions increasing, bicarbonate ions increasing, and carbonate ions decreasing. These four molecules all impact different aspects of physiology for different types of species. So for example, seagrasses will be fertilized by the extra carbon dioxide that's in the water, as may some phytoplankton by the extra bicarbonate that's in the water. Dick talked about how the decrease in carbonate ions can affect calcifiers that produce calcium carbonate shells. And then it turns out that some vertebrate species are also impacted by these changes in chemistry, likely because of an increase in bicarbonate and an increase in the hydrogen ions, the pH of the solution. So when you're thinking about acidification, it's not just the acidification, but it's the entire suite of chemical changes and how they interact with the various types of physiology of organisms. So ocean acidification is a very new science, especially for biologists like me. And in the past, when people have studied the impacts of pH on organisms, they were thinking about big pH changes, 1 unit pH change. But acidification is 0.1, 0.2, 0.3, and we don't have a deep history of doing that type of research. So there's a lot we know about physiology and how organisms interact with marine chemistry, but it's a very young field. So we can infer a lot of the impacts, but we are really in the discovery and exploration stage of how species respond to acidification. Shelled organisms are sensitive to acidification because of this carbonate ion. Well, shells, like these that you're familiar with, are made out of calcium carbonate. Okay, so calcium carbonate on the bottom left. And in a solution, these shells are always going to be balanced against the process of dissolution, and the process of accretion will be balanced. So when you decrease carbonate ions, as Dick mentioned, it pushes the pressure of this to dissolve. Okay? So these I'll pass around. This is a shell made out of calcite and a shell made out of aragonite. And just to show you what this looks like, these are geologically formed crystals of calcite and two forms of aragonite. This one made with water rounds in a cave, and this one was made in a desert. I'm going to talk to you about the impacts of the saturation state for calcium carbonate in two ways. One is this process of dissolution. So as the saturation state of a solution decreases below 1, it pushes the pressure towards dissolution. This species that I'm showing here is a pteropod, and it's one of the poster children of ocean acidification. It's a small snail that's about the size of a lentil, and instead of crawling on the bottom of surfaces like you're familiar with, it flies through the water column like Dumbo. Not only is it a beautiful, bizarre species, but it's extremely important in northern latitude food webs, both in the North Pacific and in the northern in the Arctic, but also in the Antarctic. And we have it here in Puget Sound and off of our coast. It forms an aragonitic shell that's very delicate, and it turns out this aragonitic shell is also very sensitive to changes in carbon chemistry. So these are three shells from pteropods that were exposed to 3 different conditions in our laboratory at the Northwest Fisheries Science Center for a week. So this organism lived in conditions that mimic kind of today's current atmosphere. This organism mimics future surface ocean conditions or current deep water conditions, and this indicates far future surface conditions and future deep water conditions. And what you can see is that after a week at these exposures, you had dissolution of the shell in both of the high CO2 treatments, and this occurred in a living organism. So this exemplifies the process of dissolution. One of postdocs in Dick Feely's lab, Nina Benarsik, who's doing some experiments in our lab as well, has found that pteropods that are living in the Southern Ocean are actually currently dissolving in some locations where the saturation state is low. Okay, so we can not only see this process of dissolution in the laboratory under controlled environments, but we're seeing it in the field. And that was the first incidence of collection of data showing dissolution in a wild organism. So these funny pictures show you a baby coral. Okay, so these coral were raised under different conditions in the laboratory in Bermuda. And this is, remember, saturation state for calcium carbonate. When it's above 1, remember, accretion is favored. When it's below 1, dissolution is favored. So as the baby corals settle, they produce this structure here. And what you can see is that in the high CO2 treatments, as saturation state decreased, there's less calcium carbonate there, right? This shows you the crystals that were formed by those animals. They're well-structured, big crystals here in the in the low CO2 treatment where saturation state is high, and the crystal formation breaks down in the low CO2 treatments. So this is showing the flip side of the impacts of high CO2, that it's harder for organisms to build their shells and to build their calcium carbonate structures in a high CO2 environment. So it's not just dissolution, but problems of growth of the shell. And what was interesting in this study is that Ann Cohen had some of her undergraduate assistants hand feed these baby coral. And what they found is that when the organisms were given as much food as they could possibly eat, it rescued them. when they had a lot of energy, even when they were living in a high CO2 environment, they could grow a nice big calcium carbonate structure. And that shows the biological process of how forming calcium carbonate structures takes a lot of energy. So here in the Pacific Northwest, the shellfish industry, specifically around the Pacific oyster, is a big industry. And what has happened is this artifact where we have a very closely examined species that is highly sensitive to changes in carbon chemistry. So we kind of are already paying attention to this canary. The study that was done in the Netards Bay, in the Whiskey Creek Hatchery in Netards Bay, where the hatchery realized that they had a natural experiment to explore the impacts of changes in carbon chemistry on the production of oyster in their hatchery, oyster larvae in their hatchery. And you can see here that over different days of the year, the chemistry of the water they were taking into their hatchery from the ocean varied. So it varied in pH and in this aragonite saturation state. These dots here shows the point at which they chose to spawn their oyster broodstock. So basically they spawned their oysters at different chemistries. And what they were able to do is explore the influence of that on production in the hatchery. And what they found was that production in the hatchery was lower when they spawned their oysters at lower pH, this relationship here. And they also found that the growth of the larvae when they were in their feeding stage was slower when the larvae were spawned at a lower pH. And this is the first instance of showing the direct impacts of acidification on the production of larvae in a hatchery. Just to show you what that looks like, on the left side of the pictures here are an oyster larvae at the first day of its life and the fourth day of life. And this is living, these organisms were living in a low CO2 environment, indicative of today's current atmospheric conditions. And these larvae are the same age, but they were living in a high CO2 environment. So you can see that they're not only smaller, but they're deformed. So each one of these points shows a deformity or dissolution. So as I mentioned, ocean acidification can have a lot of different impacts, not just on calcium carbonate structures and things that produce shells or coral parts, but it can influence respiration. I mentioned that CO2 can fertilize seagrass. But you have the flip side as well. So in highly energetic species like squid, their blood is the molecule in their blood that carries oxygen around is very sensitive to small changes in pH. When you put these organisms in an acidic environment, they can't get enough oxygen to their tissues. I mentioned changes in growth in calcifiers. It turns out you also have changes of growth of things like fish in high CO2 environments. I'll mention a little bit later changes in behavior, and then focus now on changes in development. When you look at the literature that's coming out on ocean acidification, a lot of it focuses on early life stages, eggs and larvae and juveniles. And that's the case for a couple reasons. Usually it's much easier to work with smaller individuals, but during the process of development, organisms are very sensitive to environmental conditions. And organisms has one chance to turn from an egg into a more complex adult. They go through major changes in morphology, especially at a lot of the marine species. And so that's one of the reasons why researchers focus on these very sensitive early life stages, because you can have big changes caused by environmental conditions. And this, just to exemplify what's going on here, you recognize our sand dollar over there? This is what a sand dollar looks like as a larvae. So big structural organizational changes are happening. And it turns out many species are sensitive to environmental conditions. As I mentioned, understanding the biological impacts of ocean acidification is a young science, and there are going to be a lot of surprises. And one of the surprises occurred with a lot of the coral, or some of the coral reef fish that have been studied in Australia. And it turns out when you raise larvae of clownfish in acidified environments, they don't behave normally. They swim towards the smell of their predators. They can't home to the smell of their native the habitat they're supposed to choose properly. And it turns out there's all sorts of behavioral changes in these organisms that are caused by a change in how a neurotransmitter functions when these individuals are raised in a high CO2 environment. So that is something people did not expect at all, that a neurotransmitter would change its ability, or that it would work differently in a high CO2 environment. So neurotransmitters are things that you're probably all very familiar with. So for example, if you take medication for depression or anxiety, that's influencing neurotransmitters. Picture of an algal bloom and some pictures of 2 harmful algal bloom species. Some work that Dave Hutchins' lab has done in the University of Southern California has found that some of the harmful algal bloom species bloom more readily in high CO2 environments and produce more toxins in high CO2 environments. So that could be a big game changer for marine ecosystems should harmful algal blooms benefit in acidified conditions. I showed this study, or put this slide up here to emphasize that not all species are going to respond to ocean acidification in the same way. So I talked about some of the negative impacts of ocean acidification on the Pacific oyster. In a similar study looking at the influence of CO2 on larval eastern oysters and Sumino oysters, Whitman Miller at the Smithsonian found that the eastern oyster was negatively impacted by acidification, but a sister species in the same genus, very closely related, was not. So the changes or differences among these species in their rate of growth influence whether they responded to CO2 in the larval stage. And so this emphasizes the point that there's going to be a lot of variability among species in how they respond to acidification. A study came out a couple weeks ago emphasizing this fact. So it was a meta-analysis where they looked at groups of corals, echinoderms, which are things like urchins, mollusks, crustaceans, which are things like crabs, and fishes. And they reviewed the literature and said, okay, what happens? Are the effects positive, negative, or neutral when these organisms were exposed to different CO2 environments in the laboratory? So anything that's red here shows a negative impact of exposure to a certain CO2 level. What you can see is that as CO2 increases in all of these groups of species, you're getting negative effects, more red. But what you can also see is that there's variability. Sometimes you'll see a negative effect, sometimes you'll see no effect at all, and sometimes even a positive effect. So there's a lot of diversity in how species are going to respond, which makes us as scientists know that we need to do a lot of research to characterize particular species. So I've talked a lot about the direct impacts of ocean acidification on specific species, but when you think about ecosystem impacts, you have to think about indirect effects. So here you can imagine that if krill, which are a major food species for whales, are impacted by acidification, that whales themselves might be affected by a drop in their prey species. Okay, so because you have linkages among species and food webs, you have to think about complex responses. So in some modeling work that I've done using a food web model at Puget Sound, I've come up with some kind of basic statements you can make about ecosystem responses to acidification. One is that species that are affected by ocean acidification will also be impacted by predator-prey interactions. So let's say one species is negatively affected by acidification, but so is its predator. You might not see a change in population as acidification increases. Similar, ocean acidification will impact species unaffected by changes in pH via predator-prey interactions. Here, again, an example, if a prey species goes down in population abundance, its predator might too. Which species are affected by ocean acidification will drive the nature of the food web response. In the modeling work that I've done, the impacts of a decline in copepod abundance were the opposite of the impacts of a decline of krill abundance on fisheries in Puget Sound. And also, ocean acidification impacts on just one or a few species can have big effects on the food web and ecosystem services. So not all species have to be sensitive to cause a change in the network, in the food web. If you have one very important species ecologically that's changed by ocean acidification, either in a positive or a negative way, that can have ripple effects through the entire community. And one of the last points that I want to make is that people are part of the food web as well. And I'll just use some little notes to cheat here and emphasize the importance of shellfish and seafood in Washington and the Pacific Northwest. So the Washington shellfish industry generates $27 million of revenue a year and 3,200 jobs. Annually, farmed shellfish from Washington comprises 85% of West Coast production. When you think about all seafood, wild seafood that comes into Washington, that seafood industry generates 42,000 jobs and contributes $1.7 billion to the gross state product. Just recreational harvest of oysters and clams generates $27 million to coastal economies. And then thinking about it beyond just money, thinking about culture, the wild geoduck fishery generates $32 million, and most of this harvest is done by the tribes. And ocean acidification is not happening in a vacuum. It's happening coincident with climate change, which is causing changes in temperature, but also changes in dissolved oxygen in oceans. In this urbanized estuary, Puget Sound, we have to think about how changes in land use are affecting nutrient runoff and pollution in marine waters as well. So all of these other stressors can impact the species response and the ecosystem response to acidification. I'll end with a few points to summarize. Dick showed that the ocean is acidifying rapidly. We know that some local species will be sensitive to ocean acidification. But we also know that biological responses to ocean acidification are variable. Some species, especially the calcifiers, do poorly in acidified environments, but some of the primary producers can do well in acidified environments. We know the impacts of ocean acidification ripple through food webs to those species that are not directly affected by the changes in chemistry. And then I touched on the fact that other stressors can exacerbate species response to acidification.